Welcome back, everyone. Today we're going to talk about fluid and electrolyte balance. And this is a continuation of our discussion on renal physiology. We also may get to the concept of concentration of urine today. So this is the section of the course where there's a lot of interplay between the cardiovascular section and the renal section, as well as the respiratory section. So there are a lot of concepts that are going to come back. And so this is one of the reasons why there's so much integration within human physiology, and it's hard to separate the systems from one another. So when we talk about fluid and electrolyte balance, it requires input, as I just mentioned, from the renal, cardiovascular, and respiratory systems. The cardiovascular response under normal circumstances is a fast response. And the reason why it's a fast response is because it is a neural control. It's controlled by the nervous system. The renal response, when we're talking about fluid and electrolytes, is a more slow response. And it's slow because it is under endocrine or neuroendocrine control. The endocrine system tends to take a while to have its effects because the hormones need to be released, and then they need to travel through the blood. They need to have their effect on the cells, compared to the nervous system, where there's a fast impulse, electrical impulse, or an action potential that stimulates a nerve, travels along the nerve, and it has its effect immediately. The endocrine responses tend to have a more sustained effect. While they are slower, they're more sustained. So when we talk about water balance in the body, water is the most abundant molecule in our body. The standard 70 kilogram male has about 42 liters of water. About 28 liters of that is in the intracellular fluid compartment. And then the rest of it is the extracellular fluid compartment, which consists of the interstitial fluid, which is the space between the cells, as well as the plasma, which is within the blood vessels. And there's about 3 liters of plasma. There's about 5 liters of blood altogether, but 2 of those liters are accounted for by the blood cell volume. Most of our water loss is through urine. There's a small amount through the GI tract, but the majority is through urine under normal circumstances. There is a thing called insensible water loss, and that is water loss that occurs through the skin and the lungs. It's called insensible because we're not aware that we're losing water. So if we think about our water loss through the lungs, every time that we breathe out, there's water vapor that is being eliminated. On a cold day, you can actually see that water vapor. It turns into the philosophical question of, on a cold day, is it still insensible if you can sense it and you can actually see it? And the answer is yeah, it's still an insensible water loss. The skin and lungs have water loss that's insensible. Any water that we lose must be replaced by the GI tract via absorption under normal circumstances. There's a small amount that is produced from all of our metabolic processes. So if you think about the breakdown of glucose, the complete catabolism of glucose, you get 6 carbon dioxide molecules per glucose molecule, and then you get 6 water molecules per glucose molecule. That small amount of water that you get from metabolic processes is nowhere near enough to regulate our fluid intake and to replace the amount that was lost. So we must replace it by absorption in the GI tract. If for some reason you can't tolerate fluids through the GI tract, there are other ways that people can get fluid. The most common of which you've probably heard about, when people are sick, they'll sometimes get fluid through an intravenous route, means into the vein. There are other ways that people can get fluid. Hypodermoclysis, which is fluid that is just under the skin, and it will be absorbed slowly. That's another way to get fluid. It's not as common, but it does work. In the event that someone is extremely sick, one of the things that happens is that their veins, most of their veins in their body will end up collapsing to a certain degree. And it can be actually very hard to get intravenous access when someone's extremely dehydrated. In those cases, there's one of two options that someone could use. They can either have a medical professional can get access through one of the very large central veins in the body, either through the jugular or through the femoral vein. Those ones tend to collapse less. But if somebody needs fluid replacement extremely quickly and you can't find a vein, you can actually replace fluid through the intraosseous route. And the intraosseous route is into bone. And the place where it's normally done is on the front of your lower leg. There's the tibia, and there will be a large needle type device that bores into the bone, and fluid is given through the bone. It is a quicker way to do it if somebody is in dire need of fluids. It's through the intraosseous route. Volume depletion, if you have volume depletion, your blood pressure drops. And when blood pressure drops, the tissues can't get adequate oxygenation. You can't get oxygen to the distant organs, and those organs are at risk of going into a hypoxic state, and they can become damaged. There are several ways in which someone can lose water. If the water loss is hypoosmotic, meaning that you're losing more water than you are solutes, there are solutes that are left behind in the fluid that's left, and you can imagine that this would raise the concentration of those fluids, the concentration of the solutes in those fluids. And our body really needs to have that fluid have a consistent amount of solutes in it. So if you end up with a large level of solute, that can raise your osmolarity and it can disrupt cell function. Many of your excitable cells can become disrupted. And if the body doesn't correct the state quickly, it could become quite dangerous. So the kidney will help us conserve water. And it can only help us conserve water. It can't replace lost fluid. So any of your lost fluid must be obtained from the external environment. That's the only efficient way to get lost fluid. Yes, you'll have a little bit through metabolic processes, but it's nowhere near enough to replace lost fluid. So if you think about water regulation and the kidney as this mug, there will be, after a certain volume of water, you can body water, this will allow for a good glomerular filtration rate. And any of this fluid will end up becoming filtered by the kidneys. The kidneys are going to conserve a lot of this volume, but there is going to be a certain amount of volume that's going to be lost in the urine. But the rest of it will get reabsorbed and it will go back to this, to the total body fluid. The only way that you can replace this water loss is by getting water from the external environment, so through the GI tract. If you don't replace the fluid that you lose, and it goes below a certain level, your glomerular filtration rate starts to fall. And that will help you conserve some of the water, but it also inhibits your ability to get rid of any toxins in the blood, and gets rid of your ability to ensure that you can remain in in a homeostatic balance of all the solutes in your blood. So it's extremely important that the volume that you lose is replaced from the external environment. And so the main point of this diagram is that the kidneys cannot restore lost volume. They can only conserve the fluid that you have. Concentration of urine is generally measured in osmolarity. If you have too much water, your urine will become very dilute and it will have a low osmolarity. If you don't have enough water, the urine will become quite concentrated. And it can become as concentrated as up to 1,200 milliosmoles. That's about 4 times the normal osmolarity of the blood. So you can actually concentrate your urine up to 4 times that amount. It can't go beyond that. You can't get urine that's more concentrated than 1,200 milliosmoles. We control urine concentration by varying the amount of water and sodium that's reabsorbed from our kidneys. And there's really 2 processes that affect the reabsorption of water or the reabsorption of sodium. And when we'll talk about each of those processes by which you can regulate those 2 things. But before we go through that, we have to look at the way that the urine that the kidney will concentrate the urine. And you have to look at the renal medulla compared to the renal cortex. And if you look at the osmolarity of the renal cortex, you look at how much solute is in there, it's about 300 milliosmoles. That 300 milliosmoles is exactly the same as our blood osmolarity. If we look at the interstitial osmolarity of the renal medulla, now deep within the core of the kidney, the osmolarity there is about 1,200 milliosmoles. That's that maximum concentrating ability of the urine, and it coincides with the maximum concentration of the interstitial fluid of the renal medulla. So we're going to look at the osmolarity of the filtrate and as it passes through the various areas of the nephron. So if we start off at the renal corpuscle, imagine that there's a renal corpuscle here. The fluid that is leaving Bowman's capsule is isoosmotic with the plasma. I need to restate that. The fluid that is leaving the glomerulus and going into Bowman's capsule, that fluid in Bowman's capsule is isoosmotic with the plasma. And so the fluid that is entering the Bowman's capsule and then into the proximal tubule is about 300 milliosmoles. As you go down into the loop of Henle, you'll actually see that there is reabsorption of water, and only water is reabsorbed in the descending part of the loop of Henle. So you can imagine that since only water is reabsorbed, the solute concentration increases. As you go down the loop of Henle, the solute concentration of the filtrate increases to that maximum amount of 1,200. Then you go through the hairpin turn of the loop of Henle and start your way on the ascending limb of the loop of Henle. And you end up with the thick ascending limb of the loop of Henle. And this region of the loop of Henle is only permeable to solutes, to ions. So there's reabsorption of sodium, chloride, and potassium here. So you've just concentrated the filtrate, but now you're taking that solute back out. And that solute that is getting taken out, it's getting taken out at quite an efficient rate, because by the time you're finished the loop of Henle, the filtrate is hypoosmotic to the plasma. It's only 100 milliosmoles. And so if you've ever learned in a course that the loop of Henle concentrates the urine, is the main factor of concentrating the urine, that's not true. It's not true, because from by the time you reach the end of the loop of Henle, you're actually less concentrated than your blood is. So it's only 100 milliosmoles. Then you go to the distal tubule. There's some fine tuning of some of the solutes. There's some acid-base regulation that happens there. And then you go into the collecting duct. And in the collecting duct, there's variable permeability to water and solutes. And that depends on your fluid level. If your fluid level is very high in your body, you need to eliminate fluids. So that collecting duct will become less permeable to water so that you can eliminate more of it. But if you are trying to conserve water, that collecting duct is going to become permeable to water, and water is going to be reabsorbed in that area. So the amount of the concentration of solute that is left over after you're finished in the collecting duct is variable. You can have a minimum concentration of urine of about 50 milliosmoles up to a maximum concentration of urine to about 1200 milliosmoles. And so the urine osmolarity depends on the reabsorption that is occurring in the collecting duct. So if your body needs to conserve water again, the distal nephron becomes permeable to water, and it's specifically in that collecting duct, and you'll end up with concentrated urine. The way that we control the water reabsorption in the kidney is through a hormone called antidiuretic hormone. It has multiple names. So antidiuretic hormone, or ADH, is one of those names. It's also known as vasopressin, and it's also known as arginine vasopressin. Both of the names, they really do speak to what the hormone does. Antidiuretic hormone means that you are going to have more reabsorption of water in the collecting duct. The other name for it, vasopressin, if you break down that word, vaso meaning blood vessel, pressors meaning constriction of the blood vessels, this hormone actually causes vasoconstriction. It causes there to be a response whereby it will boost your blood pressure. And it makes sense because this hormone is released in highest quantities when you are volume deplete, and when your intravascular volume, when the amount of fluid in your blood vessels is reduced, and that would cause your blood pressure to decrease. So secretion of this hormone will not only help regulate the reabsorption of water and increase that reabsorption of water, it's also going to allow for vasoconstriction. This hormone is secreted by the posterior pituitary. It's the posterior aspect of the pituitary gland. And it is considered a peptide hormone. So it's comprised of a sequence of amino acids. The target cells are those that are within the collecting duct. And so the way that it works is that it allows the collecting duct epithelium to become permeable to water. The water moves out of the tubule because if you think about the amount of solutes in the collecting duct, it's quite a bit less than the amount of solutes that were in the interstitium. We're going to look at an image of this. The effects of this hormone are graded. The more antidiuretic hormone you have released, the more water permeability you have. Here's a diagram looking at maximum antidiuretic hormone release compared to no antidiuretic hormone release. I just want to draw your attention to the concentration of solutes in the renal medulla, first of all. As you go deeper into the medulla, the concentration of solute increases. And so you can imagine that if you have more antidiuretic hormone, as you pass through this more and more concentrated interstitium, more and more water is going to leave the collecting duct until it goes into equilibrium with the solute concentration in the medulla. So that's why the maximum concentrating ability of the kidney is 1200 milliosmoles, because that's the maximum concentration that you get in the renal medulla. So with maximum antidiuretic hormone, you're getting the most amount of water that's being reabsorbed, and you're reaching that maximum urine concentration of 1200 milliosmoles. With absolutely no antidiuretic hormone released, there is your filtrate that is entering your collecting duct. But this collecting duct, because there's no antidiuretic hormone, it's completely impermeable to water. So even though water would like to leave, it can't. This membrane is impermeable to water. And so with no antidiuretic hormone released, your filtrate stays hypoosmotic. And it's around 100 milliosmoles for concentration. It can go down to about 50 because there is some slight reabsorption of some solutes within the collecting duct, but it's somewhere between 50 and 100 milliosmoles. That is the minimum concentrating ability of the kidney. The way that antidiuretic hormone works is the insertion of aquaporins. And this is on the tubule surface or the apical surface of the collecting duct cells. And an aquaporin is really just a membrane channel for water. The way that it works is that vasopressin is released by the posterior pituitary, and it travels through the bloodstream. And so it's indicated by these little purple squares. Vasopressin is going to bind on to a membrane receptor on the collecting duct cell. And this membrane receptor, when it binds onto the membrane receptor, it's going to signal a response inside of the cell. This is called a second messenger response. This hormone cannot enter the cell, so it just gives the message to something that can tell the cell what to do. So vasopressin binds to the membrane receptor. This membrane receptor allows for the production of a second messenger system. And this is in the form of, the second messenger is in the form of CAMP. This stands for cyclic AMP, or cyclic adenosine monophosphate. And this cyclic AMP will allow for small little storage vesicles within the cell that contain this aquaporin to make their way to the luminal side or the apical side of the collecting duct cell. And they fuse with the collecting duct cell. So now there are these channels, these channel proteins that will allow for water to leave the collecting duct. If those channels are stuck in the vesicles, there's no way that water can enter the cell and be reabsorbed. And so the more antidiuretic hormone you have, the more aquaporins are going to be introduced onto the apical surface of the collecting duct cell. There are several things that control the release of antidiuretic hormone. The most potent stimulus of antidiuretic hormone release is the osmoreceptor stimulation. And these osmoreceptors are present in the hypothalamus, which is just above the pituitary gland. These osmoreceptors are activated at a plasma osmolarity of greater than 280 milliosmoles. I did mention that a normal osmolarity is about 300. So what this indicates is that even though plasma osmolarity is normally about 300, you constantly have a certain amount of antidiuretic hormone circulating in your blood. The lower your blood osmolarity, the less antidiuretic hormone you have being released. That's the first stimulus, the osmolarity of the blood by osmoreceptor activation. Blood volume is also a stimulus of antidiuretic hormone release. It is a much less potent stimulus. It's sensed by atrial stretch receptors. So in your atria, if you have a lot of stretch, that's going to indicate that you have enough fluid volume and you don't need to release antidiuretic hormone. If your atria, if there's decreased stretch in the atria, that indicates that you have less fluid entering the heart or returning to the heart, and that will stimulate the release of antidiuretic hormone to tell your body to conserve fluid. Blood pressure is the third control of antidiuretic hormone release. It's also one of the less potent stimuli for its release, and this is sensed by the carotid and aortic baroreceptors, the same baroreceptors that allowed for the regulation of your blood pressure and bringing it back to a homeostatic level. It will also allow for the release of antidiuretic hormone. Decreased pressure is going to activate antidiuretic hormone release, and it's going to tell your body to conserve water to ensure that the blood pressure doesn't continue to drop. The control of antidiuretic hormone also follows a circadian rhythm. And what that just stands for is that there is a cyclical release throughout the day. There's more antidiuretic hormone produced at night, and therefore less urine is produced. And then less antidiuretic hormone is produced during the day, and more urine is produced. And this actually makes sense, because less urine produced at night, less waking at night, and less need to use the bathroom at night. In children with bedwetting, it's a condition called nocturnal enuresis, and it is thought to be a delay of the circadian control of antidiuretic hormone release. And so you can imagine that if there's not more antidiuretic hormone being released at night in order to produce less urine in children, they're going to produce more urine in the middle of the night, and they're going to be at higher risk of bedwetting. As this circadian rhythm matures, the bedwetting goes down. And a lot of children are successfully treated with a drug that acts exactly like antidiuretic hormone, so that they're producing less urine at night. There are other applications of antidiuretic hormone in the medical community, but they're not as common as this one. And this one's actually very infrequently discussed in physiology textbooks. So here's the control of antidiuretic hormone release: decreased blood pressure, sensed by the carotid and aortic baroreceptors. And there'll be a sensory neuron will send a signal to the hypothalamus. The hypothalamus is going to, contains neurons that synthesize vasopressin. This vasopressin is released, is sent to the posterior pituitary, where it is released. It travels through the blood to the collecting duct epithelium, and it inserts water pores on the apical membrane, those aquaporins, and it leads to increased water absorption to conserve water. And you can imagine that that would help increase your blood pressure, or at least maintain your blood pressure. Antidiuretic hormone also has the effect of constricting, vasoconstriction of your blood vessels. So you would increase your blood pressure to a degree. The decreased atrial stretch will be sensed by that atrial stretch receptor. And again, there's the sensory neuron sent to the hypothalamus. And the hypothalamus, those hypothalamic neurons will synthesize vasopressin, sent to the posterior pituitary, and then released, and the same process follows. The osmolarity being sensed of greater than 280 milliosmoles will be sensed by a hypothalamic osmoreceptor. And then there's an interneuron that gets sent to the hypothalamus from those osmoreceptors. And the same process will continue from the synthesis of vasopressin to the absorption of water, to try to ensure that you don't increase your osmolarity even greater. So you conserve the water to ensure that the osmolarity stays the same. hypothalamus, this AVP, this is an image of hypothalamus and pituitary. There's the anterior pituitary right here, and this is the posterior pituitary. AVP, this is arginine vasopressin, so that third name for antidiuretic hormone. It's made and packaged in the cell body of a neuron within the hypothalamus. Then that arginine vasopressin is transported via a vesicle down the neuronal cell. It eventually reaches the posterior pituitary where it's released. And when it's released, it enters the bloodstream. And after it enters the bloodstream, it'll make its way to the kidney, to the collecting duct cells through that process, that second messenger system that we talked about. So there's a higher medullary interstitial osmolarity, and we need that medullary interstitium to have high osmolarity. We need that because that will allow for the ability to concentrate urine to varying degrees depending on how much water you need to conserve. And the question is, what creates this high extracellular fluid osmolarity? And the answer to this is the countercurrent exchanger. This is one of the topics that I don't love talking about. I will talk about it. And you may have to go through it a few times. This is a very philosophical conversation at first, because it's really trying to explain a hypothesis for how this gradient of concentrations within the renal medullary interstitium gets created. And so we have to, in order to have this philosophical conversation, we actually have to start by talking about what is a countercurrent exchanger. And so what we mean by countercurrent are two fluids that are moving in opposite directions. So the filtrate is flowing in one direction, and the blood vessels that are surrounding the tubules of the kidney, the blood is actually flowing in the opposite direction. So you get this countercurrent that's happening. So let's look at this initial philosophical discussion. So here is a schematic of our loop of Henle. This is the renal medulla that we're looking at in the blue. And you can imagine that at the base of this loop of Henle, we're going to assume that this is the deeper part of the medulla that would have the highest concentration of solute. And so our filtrate is traveling through the loop of Henle in the direction indicated by the arrows, but the blood that's traveling through the vasa recta, or those specialized paratubular capillaries, is traveling in the opposite direction, thus creating this countercurrent. The couple of the things that you need to know is that there's varying permeability to substances as you travel along the loop of Henle. The descending limb of the loop of Henle is permeable to water, and only water. It's not permeable to solutes, so only water can leave this part of the loop of Henle. When you get up to the thick ascending limb of the loop of Henle, it is completely impermeable to water. Completely impermeable to water, but it is permeable to solutes. So solutes can be reabsorbed, or they can leave the filtrate and go into the interstitium, but water cannot follow. So when you think of the thick ascending limb, think impermeable to water. And so as you're traveling through, we're assuming at first that the concentration of the solutes that are traveling through this filtrate are the same as the plasma. And this is where our philosophical discussion starts. We're also going to assume here that the concentration of the blood is exactly the same as well. So we have to start with everything being the same. The first thing that's going to happen is that you have this filtrate that is starting to enter the thick ascending limb of the loop of Henle. When it enters the thick ascending limb of the loop of Henle, there is a transporter on these cells of the thick ascending limb that can reabsorb salt. And it can only reabsorb solutes. That's all it can do. Water can't follow. And the specific transporter that does this is something called the sodium potassium 2-chloride transporter. So there's a sodium molecule, a potassium molecule, and 2-chloride, or sodium ion, potassium ion, and 2-chloride ions that are leaving the filtrate and entering the cell. These will eventually be brought over to the interstitium. So there's going to be some solute brought to the interstitium. Water would love to follow these solutes, but it can't because water's completely impermeable. So water ends up staying within the filtrate, but you have less solute. So the osmolarity that's leaving the loop of Henle is actually less than what it was coming in. And just note that you have reabsorption of solute. Because this is a three-dimensional image, you actually have reabsorption of solute on both sides and areas of this tubule. So there's solute entering the interstitium on both sides here. So that's the first thing that's happening. And the only thing that we've seen so far is that the concentration of the solute that is leaving the loop of Henle is lower because water couldn't follow. So these solutes that have left the loop of Henle, so you can actually see that as they're being reabsorbed, the concentration is getting lower and lower until you're done in the loop of Henle. These solutes are going to be reabsorbed into the vasorecta. So you have some of these solutes that have left, and they're being reabsorbed into the blood within the vasorecta. But remember, we also have concentration here that it was about 300, and you're reabsorbing solute and only solute, no water. So the concentration is getting higher. And as you travel down within the vasorecta, it is passing by this thick ascending limb, and you just keep reabsorbing more of this solute. So the osmolarity is increasing, and it's increasing. And we're going to say for argument purposes that it goes to about 400 milliosmoles at the lowest point. Well, now it's traveling by the descending loop of Henle. Well, now that it travels by the descending loop of Henle, the permeability of the descending loop of Henle is different. It's only permeable to water. So as this higher concentration of fluid is passing by the descending loop of Henle, you can see that there's a concentration gradient, that the blood is more concentrated than the filtrate. So water is going to leave, and it's going to enter the blood that is traveling through the vasorecta. And as water is being brought into the vasorecta from the descending limb of the loop of Henle, the concentration within the blood is going down. And it goes down until it reaches equilibrium with the fluid that is in the descending limb of the loop of Henle. So this water removal will increase the filtrate osmolarity and normalize the vasorecta osmolarity. So it started off at 300, it went to 400 because it reabsorbed solute, but now it's going back to normal. And what it does to the descending limb of the loop of Henle is that as water leaves, the fluid in this filtrate is becoming more and more concentrated until it gets to about 400. So now this cycle continues, and this now higher concentration of solute is going to enter the thick ascending limb of the loop of Henle, and even more solute is reabsorbed. Even more solute is reabsorbed, and as more solute is reabsorbed and it goes into the bloodstream, the plasma osmolarity increases further to about 600 milliosmoles. The more that this means that as it passes by the descending limb of the loop of Henle, even more waters leaves the descending limb of the loop of Henle and enters the vasa recta. And this will occur until equilibrium, until the plasma osmolarity is 300 again. But so much water left that it increased the concentration of the filtrate fluid. And this process just keeps occurring until it reaches the renal interstitium reaches an osmolarity of about 1200 milliosmoles. And this is the maximum concentrating ability of the kidney. So the osmolarity of the filtrate entering the loop of Henle is still 300, and the osmolarity of the filtrate leaving the loop of Henle is hypotonic to the plasma. But as it goes down the loop of Henle, it gets concentrated, and then that solute is removed until it becomes hypoosmotic or hypotonic. You may need to go through those slides a few times, but the point of this is that the osmolarity increases as you move down the interstitium, and this creates the essential concentration gradient for the reabsorption of water in the collecting duct. Because you can imagine that this interstitium is going to have the same gradient of concentration of solute as the filtrate. And you can imagine the tubule that is next to this renal medullary interstitium is the collecting duct. And so the more antidiuretic hormone you have, the more water that's actually going to be reabsorbed in the collecting duct. And it's because of that concentration gradient that you can have, that your maximum concentration of your urine is about 1200 milliosmoles.